Good morning, everyone. Good morning, ma'am. This is my report, and I will discuss it to you about ethylene. So, what is ethylene? Ethylene gas is a major plant hormone that influences diverse processes in plant growth, development, and stress responses throughout the plant life cycle. Responses to ethylene, such as fruit ripening, are significant to agriculture. Ethylene gas with ripening process. In the ancient Egyptians, gas stimulate ripening. Lemons are the one examples, often too green to be acceptable in the market. Lemon growers used to store newly harvested lemons in sheds kept warm with kerosene stoves. With a more modern heating system, the nut turn yellow on time. The important factor in the ripening process was not heat but a small amount of ethylene gas given off by the burning kerosene. Leaf abscissions, 2% of illumination gas, dubbed 1917. In 1964, leaks of gas from street lights showed the triple response: stunting of growth, twisting of plants, and abnormal thickening of stems, inhibitions of vegetative tissues. And also, illumination gas was responsible for the horizontal growth of etholated pea seedlings. The typical symptoms of ethylene action is inhibitions of stem and root growth, leaf abscissions, horizontal growth, and plants senescence. So, this figure and this figure, can you see the process of how ethylene can produce to the plant? So, for example, for figure number 1, the ethylene insensitive mutants and the constitutive ethylene response mutant. Figure number 2, the triple response to ethylene of dark-grown Arabidopsis seedlings. Letter A, wild-type seedlings grown in the absence left or presence right of ethylene. And letter B, wild-type seedling grown in the presence of the ethylene receptor account. And letter C, close-up of the pronounced apical hook found with a triple response to ethylene. And the last is the close-up of the shortened root found with a triple response to ethylene. So we have 4 types of ethylene of dark-grown Arabidopsis seedlings. The first is wild-type seedlings. The letter B is wild-type seedlings grown in the presence. And letter C, close-up of the pronounced apical hook. And the last is close-up of the shortened root. So let's discuss about history of discovery in plants. First, the active component was ethylene. Second, plants synthesize ethylene, an endogenous growth regulator. Third, this gas ethylene was the plant hormone responsible for fruit ripening. Plant tissues ripening apples naturally produce ethylene. And about history of ethylene research. Increase in ethylene production was associated with peak in respiration during tomato ripening. Ethylene is a fruit ripening agent that acts in very small amounts. This endogenous growth regulator as a plant hormone in amounts that can reach 500 nl and g to 1H1 but is active at very low concentrations from 10 to 100 nl L to 1. Ethylene is now known to have many other functions. So let discuss about the biosynthesis and metabolism of ethylene. So ethylene is produced in higher plants, varies with the types of tissues, the plant's species, and also the stage of development. More ethylene in meristematic tissues, nodal region, dormant buds, flowers, sunsets, maturity, and ripening fruit. Precursor or methionine. Ethylene is synthesized in responses to different types of stress such as wounding, very low and very high temperatures, flooding, or drought. Treatments with other hormones, heavy metals, and attacks by pathogens. So it means dito, by biosynthesis and metabolisms, hindi siya mawawala sa isang plants or sa isang halaman. O sabihin natin vegetables. Ito ay talagang simula't simula hanggang sa ang halaman ay tumubo, andun pa rin ang tinatawag nating ethylene. Because of the tissue, plant species, and also the stage of development. So, let's discuss the ethylene biosynthesis pathway. ATP is an essential component in the synthesis of ethylene. For methionine, ATP and water are added to methionine, resulting in loss of the three phosphates and adenosine methionine. Number 2, aminocyclopropan carboxylic acid synthase facilitates the production of ACC from some. And also the oxygen is then needed in order to reoxidize ACC or AC and produce ethylene. This reaction is catalyzed by an oxidative enzyme called ethylene forming enzyme. So, parang tayo lang din yan, parang tao. Parang parehas lang sa atin sila. Kailangan din natin ng oxygen sa araw-araw. At gaya nila, halaman, kailangan din nila ng oxygen para tumubo ng maayos at masiglang halaman, mataba kung tawagin. So, itong pathway na to, dito mo masasabi kung paano mo mapapalaki ang isang halaman. Dito tutubo ng ayos. Ito yung sinasabi na the ethylene biosynthesis pathways. Hindi ko na mabasa yung figure 1 kasi medyo blurred. So, let's proceed to the next. So, this figure is showed. Parang ito lang siya. Yan, ito siya. Pathway lang din siya. Parang second example. Ethylene biosynthetic pathway. The enzymes catalyzing each steps are shown above the arrows. Each step, ibig sabihin, bawat step, dinidescribe dito kung paano ang proseso ng isang ethylene biosynthetic sa isang halaman. So, let's proceed to the another topic. The next is function of ethylene. The first function is stimulates the release of dormancy. Second, stimulates shoot and root growth and differentiation. Triple response. May have a role in adventitious root formations. Stimulates leaf and fruit abscission. Stimulates flower induction. Induction of femaleness induces flowers. Stimulate flower opening. stimulates flower and leaf senescence. It stimulates fruit ripening. Okay, this table number 1. Makikita dito ang organ or tissue and action. Makikita ang name ng organ at name din ng action ng bawat isa. So, table number 1, effects of ethylene on plant development, stimulus inhibitions. So, let's proceed to organ or tissue. Seeds, spores, and pollen. So, sa actions is germinations positive, seeds, shoots, tubercles, and bulbs, and the action of these are loss of dormancy, positive, somatic, embryos, and the action of this is embryogenesis, positive. Coleoptiles roots, growth negative, sometimes positive, modification on geotropism. Petioles, the action of this is epinesty, positive. Roots, parenchyma formations, positive, adventitious roots, positive, absorbent hair, positive. And how about the flowers? Flower induction in bromelaceous, positive. Flower differentiation in cucurbitaceous, positive. Senescence, positive, and abscissions, positive. And also the fruits. Growth, positive, ripening, positive, and the greening of citrus, positive. Stomates, closure, positive. And the last one is latissiferous, and the action of this is lattice sleeping. So, this table, the organs or tissue and the action shows or showed the function of each and the importance of its organ or tissue. So let's proceed to ethylene and fruit ripening. Fruit ripening involves a series of biochemical and structural change that make the fruit acceptable for eating. We have two major groups of fruits based on the interventions of ethylene during maturations. So, the two major groups of fruits, the first is non-climacteric fruits and the climacteric fruits. Okay, let's proceed. Okay, wait. The two major groups of fruits is the non-climacteric fruits and the climacteric fruits. So dito, makikita mo talaga yung pagkakaiba nila based on the interventions of ethylene during maturation. Once narinig mo yung non-climacteric fruits and the climacteric fruits, ang papasok agad sa isip natin ay yung makakain at hindi natin pwedeng kainin. So, let's proceed to the next. Okay. The ethylene production in C. arabica fruits during ripening. Ethylene production in coffee fruits was measured using gas chromatography. Bars represent standard errors of 3 replicates. Image below, the x-axis shows the fruit ripening stage. So dito, ito yung sinasabi na pinapakita yung x-axis ng 1, 2, and 3, 4, 5. The first is green, yellow, red, cherry, dark cherry. Different letters indicate significant differences using the LCD. So, this one, the first one is about fruit ripening stage. And the second one is about flowers. Opening flower, flower leaf drop, days. This one is 1 day, 2 days, 3 fructifications and the green fruit. And the 28 days ripening and mature red fruit. Ito yung sinasabi na stage by stage siya. Hanggang simula sa itanim at sumuhi hanggang sa bumunga. Hanggang siya sa mahinog. At ito yung last na mature red fruit. Ito yung sinasabi natin na pwede na siyang kainin dito sa part na to. So, ito na. Let's discuss about fruits. The non-climacteric and the climacteric. So, the first one is non-climacteric. What is non-climacteric? Are those whose maturations does not depend on ethylene, or such as cherry, strawberry, and pineapple. And the climacteric naman are characterized by an extraordinary increment in ethylene production which accompanies the respiratory peak during ripening, called the climacteric crisis. The next, the sharp increase of ethylene production which occurs at the initiations at the onset of the climacteric phase is the key of phenomenon, controlling the initiations of change in color, aromas, texture, and flavors. Tomato, avocado, melon, apple, pear, peach, and kiwi fruit. And dito sa climacteric, by controlling the initiations of changes in color. Ito yung part na ripening and mature red fruit. Dito nagbabago yung kulay, yung lasa, at yung texture niya. Dito sa part na to. Yan, dito. Like for example, ito. Dito makikita mo siya na light pink or medyo ma-red. Then dito, ito na siya. Yung pwede na siyang i-harvest. So dito yon yung climacteric. The next one is ethylene and climacteric. The ethylene that is released by many fruit species is coincident with the increase in respirations often associated with maturing fruit. Ethylene released by this fruit may stimulate climacteric in mature fruits located nearby. So next, yan, here, the time after harvest. Dito, it shows 1, 2, 3, 4, 5, 6, 7, 8, 9 days the apple banana. Ito yung guide or ano bang tawag dito? Ano nga ba to? Parang pinakang oras kung paano siya i-harvest. Yun. So, the next is climacteric and non-climacteric fruits. Dito, sa non-climacteric and climacteric, all of that are the list of non-climacteric, and the right side is the climacteric. So, the bell pepper, blackberries, blueberries, cacao, kasoy apple, cherry, cucumber, eggplant, grape, lemon, lime, lychee, grapefruit, olives, orange, pineapple, pomegranate, pumpkin, raspberries, strawberries, summer squash, tart cherries, and tree tomato. So the climacteric is the apple, apricot, avocado, banana, breadfruit, cherry moya, feijoa, fig, guava banana, jackfruit, kiwi fruit, mango, guava, melons, nectarine, papaya, passion fruit, peach, pear, permission, plantain, plum, sapodilla, sapot, watermelon, and quince. So, kapag nakakita kayo ng mga ganyang fruits, kahit saan, may isip nyo na, okay, this is non-climacteric. And how about the apple? Pag nakakita ko, okay, this is climacteric. So, the next one is fruit ripening. Okay. In the fruit ripening, pinapakita dito yung the cause and the effect. Okay. Let's proceed to the cause. Cause increase in membrane's permeability, which releases compartmentalized enzymes. The next, increase in protein and synthesis. Low temperature in high CO2 can reduce ethylene effects. So ethylene effects, conversions of starch to sugars via hydrolysis, synthesis of pigments, and biochemical involved in flavor. Cell wall degradations, which leads to softening. Okay, let's proceed to the several structural and biochemical changes during fruit maturations. We have 4 organoleptic qualities, such as modifications in the external aspect, texture, and flavor of fruit. So, para siyang climacteric. Okay? Same lang siya ng organoleptic qualities at climacteric. So, the change in the color of tomato fruits result from transformation of chloroplast into chromoplast, the degradations of chlorophyll, the accumulations of pigments such as cathorates and lysopenes, which are responsible for the orange and red color of the fruit, and the alterations in the textures of the fruit, the loss of fumes due to structural changes in the principal cell wall components, cellulose, hemicellulose, and pectin. Finally, the accumulations of sugars such as glucose and fructose and organic acid in vacuoles, and the production of complex volatile compounds is responsible for the aroma and flavor of the fruit. So, dito sa last, sinasabi na yung flavor of the fruit or aroma, yun yung tamis na makukuha natin kapag kinagat natin yung malalasahan natin. That's the glucose and fructose and organic acid in vacuoles. So, the next is most physical and biochemical changes are associated with activity of enzymes such as invertase and polygalacturonase, which increase during the ripening of tomato fruits, or citrate synthase and malate. Okay, so this is tomato. Leaf epineity result when ACC from the root is transported to the shoot. Meron ditong graph. Katabi ng ito, yan. Medyo malabo lang. So, in tomato and other dicots, flooding, waterlogging, or anaerobic conditions around the roots enhances the synthesis of ethylene in the shoot, leading to the epinestic response. ethylene and high concentrations of auxins induce epinasty. Auxins acts indirectly by inducing ethylene productions. Ethylene biosynthesis in the abscission zone is regulated by auxins. So, konektado sila. Yan. The shedding of leaves, fruits, flowers, and other plants organs is termed abscissions. So, ibig sabihin, the shedding of leaves, fruits, flowers ay tinatawag ding abscissions. Abscissions place in abscission layers which become morphologically and biochemically differentiated during organ development. And the next one is a model of the hormonal control of leaf abscissions describes the process in three distinct sequential phases. So dito, yung una, yung green, dark green, auxin. Yan. Makikita nyo siya dito, papunta sa tangkay. Oo nga. Then dito, medyo light green na may yellow-yellow, na papunta sa ethylene, tapos na may tangkay. Then the last one, separation layer digested because of yellowing. Ito na yon, yung lumalaglag ng kusa yung dahon niya kasi dilaw na or kung minsan tinatawag natin patay na. So, the first one is leaf maintenance phase. High oxygen from leaf reduces ethylene sensitivity of abscission zone and prevents leaf shedding. The second, shedding induction phase, a reduction oxygen from the leaf increases ethylene production and ethylene sensitivity in the abscission zone, which triggers the shedding phase. And the last one is the shedding phase. Synthesis of enzymes that hydrolyze the cell wall polysaccharides, resulting in cell separations and leaf abscissions. So the leaf abscissions. Leaf abscissions starts with abscission zone formation, triggered by photoperiod. Auxin and cytokinin production is reduced. Ethylene increases, causing an increase in cellulase synthesis. The overall plant senescence. Less is known about this process. However, some plants sense in response to completions of the reproductive process, the annuals and biennials. Synthetic ethylene. Ethipon. 2-chlorothiol-hosponic acid. When applied to plants, ethylene is generated in cryptoplasm, where psychological pH exists. So that's all, and hanggang doon lang po yung aking report. So thank you for listening.